So this is part 2 of a virus lecture given on March 25th, 2020, focusing mostly on how viruses multiply in their life cycle. And so the main points of this lecture are to go over the 5 stages common to the life cycles of all viruses, and then explain some of the basic molecular events that happen in each of these 5 stages. So as I said, the stages of a virus life cycle are split into 5 different steps, the first of which is called attachment or absorption. And so during attachment, a virus particle will bind to the host cell, which it will ultimately infect, and attach there. And attachment is one of the most important parts of a virus life cycle. The virus particle or the capsid needs to remain attached to the host cell in order to ultimately trigger an infection. And so after the virus particle attaches, the nucleocapsid, which is the capsid plus any nucleic acid, can enter into the host cell, which is where it actually goes from the outside into the cytoplasm. And then after that is the synthesis stage, where that capsid surrounding the viral genome is shed and degraded, and the host cell can start to make or synthesize viral nucleic acids, as well as viral proteins, all of the things that a virus is going to need to end up making more new virions, which is what happens during the next phase, or assembly, where these virions made up of viral proteins and viral nucleic acids self-assemble in assembly, and then are ultimately released from the cell so that they can infect other cells. So these steps are the same for all viruses. This is just to give you another idea of what this life cycle looks like for a different type of virus called a bacteriophage. And in this case, the attachment is the same. However, the entry is just the viral genome itself. The whole entire virion, including the capsid, does not enter, whereas in the previous example it did. In this case, it's just the genome that enters during entry, but the synthesis, assembly, and release are all the same. And so depending on which virus you're talking about, there are slight differences between all of these steps. But overall, the ability to attach, enter, make things, assemble, and then be released are all integral to virus multiplication and the virus life cycle. And so that first stage, that attachment stage, where the virion actually attaches to the host cell membrane, is one of the most important parts of the virus life cycle, and it's often one of the ones that is targeted most by antiviral drugs. And so in order for a virion or a virus to attach to the host cell, there are molecules that are present on the outside of the virus that must interact with molecules that are on the outside of the host cell. And those molecules on the outside of the virus are known as ligands. They can be on the outside of the capsid, as seen here on the top, or they can be on the outside of that viral envelope, depending on the virus. And they must interact with receptors on the outside of the cell membrane, which you can see here in gray. On the top, you've got a capsid that interacts with these orange receptors on the host cell membrane to attach. On the bottom, we've got triangular-shaped ligands on the viral envelope that attach to the blue receptors in order for this virus to attach. And this attachment is dictated by receptors, and the receptors on the host cell are what dictates the specificity of viral infections. And so what that means is that the specific host cells that a virus can actually infect is dictated by the presence of certain receptors. And so it's possible that if a virus can infect humans and not dogs, for example, that there are receptors on the outside of human cells that this virus could bind to that don't exist on dog cells. And receptors can also dictate specificity in an organism as well, in terms of what cell types or what tissue types a virus can infect. So it's possible that your lung tissue has different receptors that a virus can bind to than, say, your muscle tissue, which makes your virus able to infect your lung cells but not able to infect muscle. And so those receptors that are present on the outside of the host are what really dictates what a virus can attach to and ultimately affect and what it can't. And the thing that's smart about viruses, or the one smart thing that they do, is that they use receptors that are ever present on the outside of a host cell. So if that cell makes this receptor, a virus chooses one that's always going to be there on the outside. And usually that's because this receptor is extremely important to the function of the host cell. And so some examples of different receptors that viruses use to attach. On bacterial host cells, there are membrane proteins, such as lipopolysaccharides and teichoic acids, that virions can bind to in order to attach. Interestingly, virions can also bind to these external structures that are used for movement. So both pili, so you can see down here on the left, and flagella can actually have viruses bind to them and attach to their host cells in that way. And in terms of eukaryotic cells, it would be difficult for me to give many examples because each virus uses a different receptor in order to bind to a different type of eukaryotic cell. But one well-known example of a receptor on eukaryotic cells is immune system receptors, CD4 and CCR5, and they are receptors for the human immunodeficiency virus, or HIV. And you can see here in this animation, HIV virus with its spikes in red on the outside, or its ligands, are actually able to bind to receptors CD4 and the chemokine receptor in purple on the host cell, and that specific interaction allows HIV to attach to the host cell. And interestingly, and kind of relatedly, one function of antibodies is to actually block that ligand that's either present on the outside of the viral capsid or the viral envelope and prevent binding of the virus or attachment to the receptors. And so after a virus is attached, it has to get its genome into the cytoplasm somehow so that it can start to be replicated, so viral proteins can be made and new viruses generated. And in some cases, like the case of bacteriophage, the bacteriophage attaches and then basically just injects its genome directly into the cytoplasm. But the capsid that was holding this genome remains on the outside of the cell. But more often, it's not just the genome that enters the cytoplasm, it's actually the entire capsid and the genome as well. And so this is sort of a little bit easier to fathom for enveloped viruses. So an enveloped virus has a lipid membrane on the outside in blue, and that lipid membrane can fuse with the plasma membrane of the host cell, and those two lipids, once they come in close enough contact, can easily kind of just fuse or meld together, and they ultimately will release that entire enclosed capsid and the viral genome into the cytoplasm here. In order for the viral genome to be able to actually be replicated or used, however, this orange structure, which is your capsid, that protein coat, has to be removed and then degraded before the genome can be released. And so it's a little bit different when you think about naked or non-enveloped viruses. A naked virus doesn't have that nice lipid outer layer that can fuse easily with the plasma membrane of a host cell. And so what naked viruses have to do is use a mechanism called receptor-mediated endocytosis. And so the capsid of a virus will bind to receptors on the outside of the host membrane and then trigger formation of a vesicle that will basically invaginate and engulf the entire capsid and viral genome. Once that vesicle is inside of the cell, it can be taken to a structure called the endosome, where the capsid can be destroyed and uncoated, and the viral genome can be released. And so after the viruses enter, viral genes from that genome need to be expressed. And that happens during what's called the synthesis phase. And during synthesis, a virus utilizes all of the host cell machinery to make more copies of its own genome. And that varies based on whichever type of nucleic acid is used by each virus. And we're going to get more into that in the next lecture. But during the synthesis phase, it's not just nucleic acids that need to be replicated, it's also proteins. And there are two types of proteins, the early and late proteins that are synthesized during this stage. And the early proteins are proteins that function sort of helping the virus take over the cell and utilize all of that machinery. And the late proteins come in later, as the name suggests, and their functions are in the later stages of the virus life cycle. So functions in assembling the viruses, proteins that make up the capsid, and also proteins that help release of the newly made virions. And so after all of these things are made, you've got your nucleic acids and your proteins, you need to put them together to make new virions. And putting them together is the process of assembly. Nucleocapsids, which is just that genome plus capsid, need to be made, and they're made through a process called self-assembly, and they can be assembled in different parts of the cell. Depending on the virus, the self-assembly is different, and the location of assembly is also different. So I just have two examples here of a virus that's made in the cytoplasm and a virus that's made in the nucleus. Some viruses are also made in paracrystalline structures, or kind of these virus factories, or these kind of like mini organelles within the cytoplasm. And so it's a little bit easy to imagine making a naked virus that just has some kind of nucleic acid and some kind of protein. But in terms of an envelope virus, you also need to acquire that envelope from somewhere within the host cell. And usually that happens in the final stage, which is the release stage. And so some viruses are not able to be released in any other way from their host other than to kind of kill it. And they use several different enzymes, lysozyme to destroy cell membranes, holin, which pokes holes or pores in membranes and allows viruses to be released outside of the host cell and killing it in the process. However, there are a lot of viruses that are released by budding. Enveloped viruses use budding to acquire their viral envelope from the plasma membrane. And so in the case of budding, which you can see over here, and the influenza virus, you can actually see your nucleocapsid that's formed in the cytoplasm. And what happens is it will associate with the plasma membrane and bud out or form that envelope around the nucleocapsid. And it uses the plasma membrane in order to generate that envelope. And interestingly, there are some viruses that don't use either of these mechanisms for release and don't end up in the extracellular environment, but they're actually transferred from one host cell directly to the next, usually through different secretion system mechanisms. It's not important to know how that happens for the scope of this class, but it's just important to know that some viruses have to kill their hosts, some are able to leave their host intact and just bud out and acquire their viral envelopes like that, and some viruses can be transferred directly from one host to the next one.